Good day. Today we are going to look at how Mendel actually established the foundations of genetics, the kind of experiments he was conducting. So he mostly worked with pea plants. That's why you see here Mendel and pea plants. Before we go into that, let's look at pollination with flowering plants, or the plants that produce flowers, the common ones that you know around. Pollination is the transfer of pollen grains, and remember, our pollen grains contain sperm cells, plant sperm cells. Transfer of pollen grains from the male anther of a flower to the female stigma of the flower. The female stigma is connected to the style, so it forms the female wall of the flower. It's connected to the style, which leads to the ovary, inside of which we find ovules. These ovules represent the egg cells. Therefore, pollination is a transfer of pollen grains from the anthers, which form part of the male wall as well, which is made up of the anther and the filament. Pollen grains will be transported from the anther, either by wind or by insects or different agents of pollination, to the stigma, and the sperm cells will start digging their way through to reach the ovules where they will fertilize the ovules. Now, this could happen in two major ways. It's either the pollen grains are being transferred from the anther to the stigma of the same flower. In that case, we'll refer to it as self-pollination. Or the pollen grains are transferred from the anther, this time to the stigma of an entirely different flower growing on a different plant. You can see the plants here are different. If this happens this way, like in C here, we'll therefore refer to it as cross-pollination, where the plant must be of the same species or more or less closely related species. Therefore, a new individual will be born from that. That will be sexual reproduction. In this case, we are looking at sexual reproduction because it involves gametes, production and fusion of gametes. The gametes here are the sperm cells and the ovules. So with that in mind, let's look at our next slide here. This is the work of Gregor Mendel. Gregor Mendel was always crossing pea plants, different parents of pea plants, and trying to investigate or check the pattern of inheritance in the different generations. For him to do these crosses successfully, he needed to have entirely different plants, let's say plant A and another plant B. Both are pea plants, that's the same species. But we should understand, if we do self-pollination, it's as if it's an individual plant which is producing its own offsprings from its own genetic makeup. Therefore, the offsprings will most likely be almost exactly like the parent. They will have more or less the same characteristics like the parent. But that's not what Mendel was interested in. Remember, at Mendel's time, people used to have the perception that if a tall person, for example, has a child with a short partner, let's say a tall man and a short woman having a child together, people used to think that the characteristics that will be expressed in the offspring or the child will be a blend of the characteristics expressed in the parents. Therefore, tall plus short, they will think that the child will be medium-sized. So that's the kind of thing that Mendel wanted to prove. And that's why he had to get plants that are completely different, not the same flower of plant self-pollinating itself. In a nutshell, Mendel was most interested in cross-pollination. And for him to achieve that, if you notice, a flower on its own has more or less everything that it needs for pollination. It has the anther, which will supply the pollen grains. It has the stigma, which have to receive the pollen grains. So if he had to keep a flower the way it is, then it won't necessarily work. So what Mendel had to do was, he had first to get one flower, he had to cut, you see here, he's cutting out the anthers of the flower. In this way, he is putting away all the pollen grains, because the pollen grains are contained in the anthers. He's putting away all the pollen grains. He only needs the stigma of that flower to ensure that self-pollination will never be able to occur here. Why? Because this flower has no anthers anymore. It has no male parts anymore. It has only the female part, which is the stigma. So this flower cannot self-pollinate. Therefore, he artificially made cross-pollination possible. A flower whose anthers were removed received pollen grains from another flower with anthers, which was growing on a different plant, but of the same species, both pea plants, which was growing on a different plant. So he transferred those pollen grains artificially, and artificially did or conducted cross-pollination. In that way, he was taking genetic material from one plant, dropping it on another plant. The sperm cells will dig through and meet the genetic material that's found in the ovules of the plant whose anthers were being taken out. In that way, it is just as if we are crossing. If I'm saying this is plant A and this is plant B, it's more or less now as if we are crossing plant A with plant B. And we're trying to find out what will happen with the offspring, what kind of characteristics they will express. That's the kind of thing Mendel was doing. Now, Mendel mostly used pea plants for several reasons. The most prominent reason was the fact that pea plants exhibited at least or about 7 contrasting characteristics. It means that these were characteristics that were very much observable, easy to observe, characteristics that were different from one another. For example, flower color. He could have some pea plants with purple flower, and we clearly see that purple is different from white, which could be from another plant. Again, height. Pea plants were always either tall or short, and that's clearly very observable. Seed color, another characteristic. Some seeds were yellow. Some pea plants produced seeds that were yellow, while others produced seeds that were green. Very, very much observable. Seed shape. Some pea plants produced seeds that were round, while others produced seeds that were wrinkled. Pod color. Some pea plants produced green pods, while others produced yellow pods. Pod shape. Some pea plants produce inflated, otherwise full pods, while others produce constricted or flat pods. They were flattened. And flower position. Some pea plants produce flowers that were found at the ends or towards the ends of the plant, around the middle of the plant. That was axial, while others produce flowers that were found around the ends of the plant. The flowers were terminal flowers. So we have here 7 characteristics, and we see that each one is entirely visibly very different from the other. That's why it was easier for him to work with pea plants. Now, when Mendel did his crosses, say he took 2 parents, Mendel always started with what we call pure braids. We also call them, pure braids. He always started with plants that were pure braids. Or we also call them true braids. True braids. So, the other reasons I forgot to mention, the other reasons why Mendel chose to work with pea plants was the fact that they reproduced very quickly, comparatively to how humans, for example, reproduce. It was easy for him to establish new generations of plants, to see the expressions of the different combinations of genes that he would have suspected in each plant, to see how they express themselves in the phenotypes of the offspring. So pea plants are seasonal plants and the seasons are short. Hence, we'll get new offsprings or new generations in a very short time. Imagine him working with human beings. For a human child to be born, let's say a male child, when that child is born, he will have to wait for maybe 14 years until that boy reaches puberty before he can also give rise to his own children, and then Mendel will have to be looking at the different generations. It takes very long. Before Mendel could reach, say, 5 generations, it could have taken all of his lifespan. So it wasn't that easy to work with humans. Working with pea plants was much shorter time spent in between the generations. That's why he also chose to work with pea plants. There are many other reasons. I won't dwell on that, unfortunately. Now, I was saying, traditionally, we would like to always start with our letter T. I will emphasize on this later on. For Mendel, he was always working with pure breeds of plants. It means plants that were homozygous for a particular characteristic. So he will cross 2 parents. Each of them you'll see here. I already explained the terms homozygous and heterozygous. You see that all of these are homozygous, and then he made those kind of crosses. That's how he used to work with plants. Mendel did several experiments, more than 1,000 experiments, and observed more than 1,000 generations of plants. And he did that to increase the reliability of his experiments, to make sure that the information he was gathering were very trustworthy. Okay, so with that, he therefore came to certain realizations and established certain laws. We'll investigate the laws now. So here we are going to look at, or we are looking at the laws of Mendel. Okay, we are looking at the laws of Mendel. Mendel's first law is a law of segregation. Segregation also means to separate. Okay. Segregation also means to separate. The first law states that there are 2 genes. I drew here chromosomes, homologous chromosomes. And remember, in a pair of homologous chromosomes, one is always from the father and the other is always from the mother. And chromosomes are basically made up of DNA, which are also made up of portions that we refer to as genes. I've written letters here, T and C, that will represent the genes that we'll be talking about. Back then, Mendel wasn't using the word genes because no one had coined that word before. He used to call them inheritance factors. Okay, he used to call them inheritance factors or factors of inheritance. And today we call them simply genes. Now, Mendel's first law, it states that there are two genes or alleles, we already explained alleles as well, controlling a given characteristic, and these alleles separate and go to different gametes when a parent reproduces. Now, here Mendel is actually talking about meiosis. During meiosis, we'll see chromosomes that align themselves at the equator, and I'm talking about meiosis because meiosis is the type of cell division that leads to the formation or production of gametes. So, during anaphase, we'll see chromosomes actually separating and going to different poles. If these chromosomes had aligned themselves at the equator next to one another, as homologous chromosomes do during meiosis, they pair up, especially at meiosis 1. That's metaphase 1. If they had aligned themselves at the equator, during anaphase, they will separate. That's the separation Mendel is talking about. And notice here, as the chromosomes are separating, it is also the genes or the alleles that are actually separating. Or otherwise, as the alleles are separating, it is actually the chromosomes that are separating. In this case here, I've considered just one gene, the gene for height, but in one chromosome, there are thousands of genes. But our gene of interest here is the one for height. That's why I'm only putting that one. As this one is separating, other genes are as well separating. And that's what happens so that one gamete will inherit only one copy of each gene. We see this with our monohybrid crosses. So that is Mendel's first law. When parents are reproducing, before you reproduce, you produce first gametes. Say this is a male parent. This male parent needs to produce sperm cells. The sperm cells will carry a copy of each gene as per our diagram. And for that to happen, during meiosis, the chromosomes will have to separate. Remember, during meiosis, we're moving from 2n to end up with the gametes having n. That's why in the complete set of chromosomes here, you see the alleles are still together. The chromosomes are still together. Everything here is 2n. At the end of meiosis, we are separating so that everything comes down to n. That was the first law. Mendel's second law, the law of dominance. Now, Mendel did his experiments, and each time he crossed with purebreds, he realized something. Let's go back to our purebreds cross here. This is what Mendel realized. He took two parents of purebreds. One was tall, purebred, it means homozygous, tall, and the other was short, it means homozygous short. So when he crossed them, first they will have to segregate. The alleles here will segregate so that the gametes are produced. And when they were crossed, the offspring that were born, this is going to be the first generation offspring. This is the first generation offspring. There are four possibilities here, or four possible outcomes. Mendel realized that all the offspring, when he looked at them physically, they all had the characteristic of just one of the parents in terms of height, and that characteristic was tallness. They were all tall, just like one of their parents. But the characteristic of the second parent was never represented. When he always repeated with these two parents, the outcome was always the same. The offspring were always all tall. He then thought to himself, What if I take two of the offspring and cross them together? What will happen? Let's see what happened. So Mendel took two offsprings. Remember the genotypes of the offspring. That is now our F1. And these offspring were all, they were actually all tall. Remember, our offspring were all tall. So he crossed two offsprings together, both tall this time. And in their genotype, which was not visible physically, all we could see was tall, the genotype showed that they were all heterozygous. Now, after meiosis or after segregation, this is what we had. When fertilization took place, this is the outcome that we are having. Okay, dominant always comes first. Okay, then we have that. Now, when Mendel crossed the first generation offspring, they also became parents. Therefore, we'll call them the P2 because that is second parents. He realized that out of 4 offspring, always 3 of them, that gives us a ratio of 3:1. 3 of them were always having the characteristic of the very first parent, which was tall. And to his surprise, one of the offspring was actually short. Then remember, in the first generation, none of the offsprings was short. They were all actually tall. But surprisingly, we see the trait of one of the original, the first parents showing up. The short trait there showing up. So Mendel asked himself the question, or it would have sounded that the first time that the parents were crossed, when we still had the pure braids, you would have felt as if the trait for shortness had totally disappeared or was non-existent. But surprisingly, when it shows up again, it brought to Mendel one realization. He realized that the trait for shortness had not disappeared completely. It was rather being concealed or hidden in the first generation offspring. And that is why only under certain conditions would that trait show up itself again. So he therefore established his second law after doing this experiment many times to increase the reliability. He established the second point, or the second law, which stated that if two alleles are different, this is it here, heterozygous. If two alleles are different, e.g. TT, this is heterozygous, the trait associated with only one of these will be visible, while the other trait will be hidden. Therefore, the trait that was always showing up or being visible in combination of the two different alleles, he called that trait the dominant trait. Otherwise, the dominant allele. And he called the trait that would be hidden in the presence of the dominant one, he called it the recessive allele. It tells us that The recessive allele doesn't completely disappear, doesn't become absent in the gene pool, no. It is just hidden, concealed by the dominant one. So to say, as they usually say, when the cats are around, the mice hide themselves. It is only when the cats are absent that the mice come out to play. Therefore, the reason why the recessive trait, shortness, reappeared was because in the second generation offspring, there was a combination of two recessive alleles, the small t's. It is this that created the only condition for a recessive trait to be expressed in the final phenotype. Therefore, for a recessive trait to be expressed in the final phenotype, the alleles for that recessive trait must be in combination with one another or be in a homozygous state, or the dominant allele must be completely absent. Therefore, the recessive trait can express itself in the final phenotype. That was Mendel's second law. You can still see it being expressed here. It tells us here that yellow is dominant. You'll see I have a pea plant producing green seeds crossed with another pea plant producing yellow seeds. All the offspring are heterozygous for the condition, but phenotypically they are all yellow, expressing the characteristic of just one parent. It means yellow here is dominant. Mendel's third law, the law of independent assortment. Mendel went on to do other experiments which involved the observation of the pattern of inheritance of not only one trait. We've been dealing, for example, with just one trait, that was height. This time Mendel observed two traits at the same time. For example, he could observe seed shape, like here, and seed color. In this diagram here, we have two possibilities for each trait. For seed shape, it could be either round or wrinkled. And for seed color, it could either be yellow or green. So we are observing two characteristics here. And Mendel did this experiment and came up with his third law, which states that different characteristics are passed on to the offspring independently. That word independently is what makes us call it independent assortment, independent mixing. Okay, characteristics are passed on to the offspring independently of one another, and so a combination of traits or genes or alleles in the offspring do not always match the combinations in the parents. I'll explain this. In other words, what Mendel meant was genes from one parent are not bound by one another. They are free to move about at random independently of one another. Let's make this practical. Now, I've prepared here two chromosomes, one from each parent, and my chromosomes this time have alleles for two different characteristics. Say, for example, this one is for height. I have dominant tall, recessive short. And I have here for fruit color, it could be red fruit, which is dominant, and green fruit, recessive. Okay. This is one individual, and these are the kind of chromosomes he has in his karyotype. The chromosomes here are homologous chromosomes. So what Mendel meant is, during meiosis, during the time of gamete production, the traits, the genes, or the alleles separate and go to to each gamete independently or not bound to one another. If we want to form gametes from here, meiosis always halves the number of chromosomes, or it always halves the alleles or the genes. One obvious gamete that will form from here would be a combination of these two alleles here. This will give rise to capital T, capital R. Another obvious one here would give rise to small t, small r. So these are two gametes. Now let's apply independent assortment. When we say something is independent, it means it has a free will. It acts the way it wants, free and not bound to any rule or anyone. Therefore, let's say this allele here says to this guy, it's not because we all come from the same chromosome or from the same place that we will be always together in the gametes. What if I can go and meet this other one from this other chromosome? I will form a different gamete, which will be T, R. That is my gamete. Now we have three gametes. 1, 2, 3. This one also says, it's not obliged that I be with you. What if I also go away and form another gamete with that other one from the other chromosome? Because I am independent, because I am free, we need to mix at a random. Then I'll also form capital T, small r. This is another gamete. We now have 4 gametes. So this is how independent assortment occurs. And to write it out simply, what we have here is, this is one individual. Remember, both chromosomes belong to just one individual. During the time of gamete production, these individuals' chromosomes will have to independently assort themselves. Otherwise, the alleles will have to independently assort themselves. We can represent it this way. This one says, I could be with this one, and I give TR, capital T, capital R. This is one gamete. I'm sure we have that gamete somewhere here. So this one has been taken care of. You could also say, I'm not obliged to be with just that big R. What if I also go with the small r here? Then I'll have capital T and small r. This is another gamete. I'm sure we already also had it here. So I could say this one as well. This one as well has been taken care of. Okay. And this other T says, myself, I can also combine with another R somewhere to produce my own gamete. And I'll write small t, capital R, and I form a new gamete. I'm sure we have that gamete somewhere here. So I can cancel that. It has been taken care of. Again, the small t could say, I could also go and form another bond somewhere here, and I have small t, small r. I'm sure I'll also have this gamete somewhere here. It's already represented as well. I can also roll it out. So what I've done here, I've explained to you what happens physically, naturally with the chromosomes. But when we are writing out genetics, or we're representing this genetically on paper, we won't need to be drawing cumbersome diagrams like the ones of the chromosomes we have here. We make everything simple. Okay, we're making everything simple. We have produced 4 different gametes. Here they are, out of independent assortment. Notice that each gamete contains two different letters, not the same letter in capital and small letters, but two totally different letters. It tells us that if you find under independent assortment yourself writing something like this, it means something is already totally wrong in terms of gametes. And notice again that from the parent here, there were 4 alleles. The ones I was considering, there were 4 of them. 1, 2, 3, 4. So these 4 would represent my 2n. And when they divide through meiosis, I'm expecting to have n. So there were 4 here, 4 alleles. Now look at each gamete. In each gamete, there is just 2, 2, 2, 2. Therefore, my n here will be 2 because we all know that 4 divided by 2 is equal to 2. 2n divided by 2 is equal to n. So my 2 here represents n, which is the number of chromosomes in the gametes. Therefore, we can conclusively say that meiosis took place here. This is still meiosis. Please don't confuse this with fertilization just because we're seeing this kind of crossing here. Okay. So that was it for the laws of Mendel. Thank you very much for attending. Hope to see you next class.